we're ready to get started here. Thanks, everybody. I have the amazing pleasure of moderating what I would call a super panel. We have three talented, experienced, wise people that are going to tell you about their work. I don't want to steal a lot of their time, but we have Virginia Rauh from the Columbia University Medical Center, Routt Reigart, emeritus from the Medical University of South Carolina, and Chen, who's Icahn School of Medicine at Mount Sinai. We couldn't ask for a better way to learn about pesticides this morning, so I'm just going to let them have at it. I think, Virginia, you're up first. Thank you. If you just press the down arrow.
Good morning, everyone. I'm going to try to get us back on time a little bit, but I don't want to shortchange everything. So I think that most of you have a pretty good background in this area, and I think you could probably have made up this slide yourself. Why we're concerned about human health effects is obvious to most of you in the room. I'm particularly focused on the prevalence of the chemicals that are in the environment and the vulnerability of the developing brain. I want to say also that EPA in its past has done a terrific job in helping to regulate some of these pesticides, but as all of you know, things have slowed down a little bit on that front, and I think we have to work toward getting things back on track. So for those of you who are almost as old as I am, this is 1945 Jones Beach, which is a New York City landmark, and that's a DDT truck. I think I swiped this slide from Mary Wolfe a number of years ago, but it's true that we had very little awareness of what the hazards were. And now, things aren't much better in some ways. Maybe the bad actors have changed, but we have a highly prevalent use in particular of chlorpyrifos, which is an organophosphate pesticide. So in many parts of the world, this scene is commonplace. And I'm going to say a few things about the Hawaii situation. Waimea town is on the so-called pristine island of Kauai in the state of Hawaii. And that's where a lot of the test fields were located or are located on the west side of the island. The Hawaii story is a good one, but they endured years and years of exposure. So just a little bit about why we would talk about this particular chemical. This is a broad-band organophosphate insecticide. It was banned in 2000-2001 for indoor residential use. It remains on the market and is widely used for agricultural purposes and for many purposes, as you can see in the bullet. So the Columbia Children's Center, which has been directed by Ricky Pereira, we started the center in 1997, 1998, and we have a number of ambient exposures we've looked at. The pesticides have been quite important. The children are now, the oldest children are turning 20 years of age, so we have some pretty good long-term health outcome information. For chlorpyrifos in particular, it's a complex chemical in the sense that it may have multiple paths of hazard. It was originally picked up because it was an acetylcholinesterase inhibitor. There may be other mechanisms by which this particular chemical helps and harms the developing brain. And I know that it was noted last night in one of the sessions that it's probably, even at very low dose effects, has a measurable effect. So I just want to say a couple of things about the Children's Center for Environmental Health at Columbia. This cohort is a northern Manhattan cohort, Washington Heights, the South Bronx, Central Harlem, and we've been following the children, as I mentioned, picking the moms up during pregnancy for almost 20 years. We've had a number of findings over the years, and a number of people that I can't possibly acknowledge have been affiliated with this work, including the wonderful research assistants who are of the community and who've been with us, some of them for almost 20 years. So we had early findings that this particular chemical was associated with decrements in birth weight to the tune of about 150 grams, which is not much less than the decrement in birth weight associated with maternal active smoking in pregnancy, which is about 200 grams. We had findings early on in the motor and cognition area when the children were little, and this was followed up by assessments every couple of years. We published again at the 7-year point where we were using the Wechsler scales to assess cognition. I want to say a few words about this because I think it's very important in terms of recognizing the epidemiology. There was an issue in environmental health perspectives, and all 3 groups that were looking at this particular chemical at the age of 7 years of age as the follow-up had very, very similar findings. And the journal, I think, was agreeable enough to publish all 3 papers together. What's important about this is in the world of epidemiology, and we take a lot of hits about this because we don't do clinical trials, it's observational. But in the world of epidemiology, corroboration is obtained in a number of ways. And these 3 studies, I think, are a really good example of how this can be shown. First of all, they're in very different places, although the Mount Sinai sample was also in New York City. Different populations. They used very different biomarkers of exposure, urinary metabolites as well as a blood biomarker. And I think it's really important that 2 of the populations were urban and 1 of them was rural, agricultural. So in the world of corroboration, despite what the agrochemical industry might be telling us today, this is a very powerful statement. The 3 studies at very similar findings at the age of 7 years. And these are what I would call mild to moderate cognitive deficits. So what we did to look at the children a little bit further along, we did some complex factor analysis to determine what exactly the children looked like who had been highly exposed to chlorpyrifos in our sample. And keep in mind, this is an indoor urban sample, and you're aware, those of you who are from New York City, that chlorpyrifos was used primarily to control for pest infestations. So most of us have seen a cockroach, and a lot of the housing in New York City contains cockroaches in need of some sort of a remediation, and chlorpyrifos was particularly effective. But this is what we found, and this again is sort of more of a neuropsychological perspective in looking at different domains of kids' development. These children were somewhere between the ages of 9 and 12 when they were measured. Take a look at the red line. This is the group that contained the highly exposed chlorpyrifos children. What's really important here is to see which areas these children were somewhat different from the rest of the population. So we're not looking at ADHD-type kids with a lot of impulsivity. We're considering that these children primarily have some attentional disorders. But what really, really sticks out is the finger dexterity. And that's where the red line plunges. And you can see that it departs from the other. Now, what is finger dexterity? The neurologists do these kind of little tests. And it's perhaps not so major in terms of, whether or not a child would be capable of playing the cello. Who knows? But the point is, what does it indicate? What are we talking about here? And this is something that actually hasn't been discussed very much. We did an initial look at the children when they were between 5 and 9 to take a look and see what some of their structural brain elements looked like. And I've summarized this in the next 2 slides with 2 explanatory slides just to show you a little bit about the persistent brain developmental differences. So we found overall that among the highly exposed children, it wasn't so much that they had smaller brains as they had volumetric volume reductions in certain regions of the brain. And we found that these particular regions correspond to certain functions. And this is sort of what it looks like. This cartoon is simplified a little bit in the sense that I've put the labels as to what function maps to those sections of the brain. And keep in mind, in neuroimaging, the brain is a very plastic organ, meaning that it can recuperate. And there are many ways that the brain can result in effective functioning. So it doesn't map exactly in certain parts of the brain to function, but this is a good summary about the areas in which we saw either unusually small regions of the brain or unusually what we call perturbations in size, enlargements, abnormal enlargements. And where you see, this is not an fMRI where things light up. This is an MRI where it shows the colors are simply an artifact put on here to show you where the differences between the high and low exposed kids lie. So you can actually see that there were differences in areas that map to attention, receptive language, executive function, reward and emotion, language, and receptive language in particular. The other, there are many brain findings, but the other one that has some intuitive appeal, I think, and folks can relate to, is there were areas of the brain, many areas of the brain, where we noted cortical thinning. And again, it's those purple-pink areas that are really showing up. And these are really, distributed in both hemispheres across the cortex, and it shows that we did have cortical thinning in the most highly exposed children. So just to summarize what I've said in the last couple of minutes, even at very low levels, which are standard usage right now in the agricultural community, the exposure to chlorpyrifos was associated with structural changes in the developing brain. And we have some evidence that these are persistent. We're talking about a prenatal exposure. So the fact that we still find differences in the structure of the brain at the age of 11, I think is quite interesting. Now, what we decided to do is to take a look and follow up this motor movement problem that we were seeing. Now, there's some very interesting data from the past among adults who had high occupational exposures to chlorpyrifos and then developed Parkinson's disease. We wondered if perhaps we were seeing in our early exposed children who didn't have such highly dramatic exposures, if we were seeing the very beginning of a sort of a neurodevelopmental trajectory that might eventually lead to neurodegeneration. And as many of you know, Parkinson's disease is the second most common neurodegenerative disorder currently, next to Alzheimer's disease. So why we would look for Parkinsonism? We know that this exposure has been associated with Parkinsonism in adults with high exposures. We know that we have continued exposure and we have increasing rates of Parkinson's disease, certainly in the United States, and there is some early evidence that we may be seeing some indicators. So when we're looking for Parkinson's disease, we're not actually expecting in 20-year-olds that we're going to see frank clinical Parkinson's disease. It tends to be late onset. There are cases, lots of examples of early onset, but typically not in the age group we're looking at. So what might we be looking for that would actually give us clues that maybe we are setting a toxic process into motion? So we look at a number of symptoms and early indicators, and here we are collaborating with the neurologists at Columbia University Medical Center to see if our children are showing any signs that may be considered premotor, preclinical. And I would say that of the findings, probably the tremor was the most marked in the sense that these children have what I would call a very mild tremor. These are the areas we're looking at. As some of you may know, also in Parkinson's disease, it's sort of predated by a loss of the sense of smell, hyposmia. So we're looking in a lot of different areas to see if we can find any early indications. We have found, without belaboring this part of the brain, which is an area of the brain where Parkinson's patients actually have a cell death, apoptosis. Usually post-mortem findings, you can see that this area of the brain is depleted of cells. It looks like our cells have been are starting to decline. So where are we headed in terms of future directions? Well, we have some evidence, growing evidence for the developmental toxicity of organophosphates from multiple different studies in multiple different populations. In the absence of federal regulation, as you all know, many states are considering legislation that would ban chlorpyrifos and other organophosphates. I had the pleasure of going out to Hawaii. This is one of the successes, one of the early successes, to speak with the legislature about the science, and then had the high pleasure of speaking with the governor as to why I thought he should sign this bill. So in Hawaii, which is the first state to effectively ban chlorpyrifos, it was a big effort that took years of advocates to actually raise awareness. That slide early on that I showed about the Waimea community was pivotal in that the test fields that were used by all the industries were essentially located right near populations of indigenous Hawaiian residents and school children. So I would say that there is we have the potential to do something here, bringing the science to the policymakers. And I think it's wonderful to be here with you this morning because I think it's another step in the process. Thanks.
Thank you so much. Next, we have Routt Reigart. Do you have a PowerPoint here, Routt?
Yes, it's on the desktop.
Okay, can we get?
I think I can do it. Okay. There it is.
Okay, there we go. Great.
Okay. I can't neglect pointing out that Beyond Pesticides was very active in the Hawaii effort, and we're still active in Hawaii, and actually have an employee working in Hawaii. So we're excited about that effort. I'm going to spend a little time talking about a paper that was published in January. The authors are me and Dr. Roberts and Erin Dolly, who's our research assistant. And I'm not going to give you everything that's in the paper because it took us many months to write this paper, and it would take me many months to describe everything that's in it. But I'd like to say a few things about it. First off, we're invited to do this review, and then we're limited to 4,500 words, which anyone that knows how to count words in a paper is a pretty short paper. But we're asked to talk about children and pesticides. We decided to do it a little different from what Dr. Rauh has just described to you, which is going from the chemicals to the effects. But we picked two very important diseases in childhood, which are autism spectrum disorder and attention disorder hyperactivity. It's important, and I think all of you know, but I'm going to emphasize it again, to remember that the origin of these diseases is prenatal and very early in life, because we begin to see the symptoms very early in life, and therefore something is happening during critical stages of development. And you heard Dr. Rauh talk about morphogenesis, synaptogenesis, these events that are occurring in the developing nervous system. We also wanted to go into sort of an integrated approach, looking at the sort of multiple lines of evidence rather than doing what you read in the New York Times, which is someone says, oh, coffee is good for you, coffee is bad for you, it's the latest little bit of evidence, or you should eat eggs, you shouldn't eat eggs. What we wanted is sort of put it all together in a logical framework for biological plausibility now. And just to remind all of you that correlation doesn't necessarily mean causation. None of us think that Nicolas Cage causes people to drown into a pool. But you can see there's a very tight correlation here. So correlation itself doesn't prove causation. And again, you heard from Dr. Rauh that when you have multiple different studies, it starts to become more convincing to us as scientists. And this is a very old set of criteria that has been used to evaluate epidemiologic research. The important things is how strong is the association? Does increased dose give greater response? Does the dose or whatever it is occur before the event rather than simultaneously? Dr. Rauh talked about the consistency of findings, and you need a biologically plausible mechanism. Again, what you heard from Dr. Rauh about change in the structure of the brain in response to chlorpyrifos is a biologically plausible mechanism for something to occur, and then so these are the sort of things that I look at and all scientists look at as evidence that there's really something going on here, and it's not just drowning in movies. So we chose these two entities because they're actually very important. Both ADHD and autism spectrum disorder are rising dramatically in prevalence. Again, for both of them, unfortunately, boys get it more than girls. As you can see, the present estimates of prevalence are very high. The number you always hear about ASD is it's 1 in 69 children. The important thing in this rise in these diseases is it almost can't simply be a genetic change because the genome doesn't change that fast. Now, in recent years, we've begun to recognize that you can alter genetic expression by so-called epigenetic events. So there definitely is an interaction between genetics and the environment in these increases in prevalence. And as I said a minute ago, we really need something to happen very early in the life cycle, either in utero or shortly after birth. So what happened? Well, we did a sort of extensive search and found 249 papers related to the interaction between pesticides and these entities. Of those, 115 were human studies. There were 15 review articles that weren't very useful to us because we wanted the original research. And of the human studies, 95 of the 115 reported associations between pesticides and ASD. And like you heard from Dr. Rauh, when you have multiple studies showing associations, they become more believable. And there were 5, if you add, I know this is a little confusing, if you add 15 to 95 to 5, you get the 115 human studies. And only 5 concluded that there was no association. So our logic in approaching this is there are a variety of lines of evidence, things done in vitro in the test tube, animal studies, and then human studies, which, as you've heard, are primarily epidemiologically. And our basic logic was that concordance between experimental studies and epidemiology supports causation in humans more strongly than any single line of investigation, and this supports a biologically plausible mechanism like I talked about in the Bradford Hill. So these are just a few of the findings. You got to read the paper. You won't get it till you read the whole paper. But ADHD in mice and rats cause behaviors similar to ADHD with a variety of organophosphates, pyrethroids, and endosulfan. Again, it's early exposure, prenatal and early life. In vitro studies showed alteration in the dopaminergic, I can't ever say that word, studies. So this is a chemical change. And the one that I actually found most amusing is that one of the neonicotinoids, imidacloprid, makes fruit flies hyperactive. Y'all should enjoy that one. And there's looking in vitro to certain brain receptors in neurons, these same chemicals. So here we have, for ADHD, changes in animals, changes in fruit flies or animals, I guess, and biochemical alterations. We selected among the ADHD human studies 12 key publications. 5 of them were cross-sectional, that is looking at kids at a single time and relating to exposure. 5 prospective cohorts, that is where someone was looking at kids going forward. 1 so-called case control. The important thing among these 12 publications, there were multiple pesticides and specifically organophosphates and pyrethroids that seemed to be related to the reduction of ADHD in children. In the laboratory for autism spectrum disorder, there were a variety of studies in cell culture, including human placenta cells and cortical neurons, brain neurons. In the mouse, where alterations were produced by these chemicals. And I mentioned a minute ago, multiple pesticides have been shown to cause methylation of DNA, which is one of the major mechanisms for so-called epigenetic changes, which can be passed from generation to generation. And again, there is a mouse strain, which I won't go into because it's very technical, that have behaviors that mimic ASD, autism spectrum disorder, and this mouse strain had increased rates of this ASD analog by exposure to a whole variety of pesticides. That's the laboratory. In the human, we found 10 key studies that we summarized, whole variety of cohorts, and ASD was associated with multiple pesticides, including particularly organophosphates and organochlorines. Okay, that's the story. Basically, we looked at many, many studies, not just the latest epidemiology. We found that in the laboratory, in animals or cell cultures, a variety of systems, we can find things that mimic these two important diseases, autism spectrum disorder and ADHD. And we found a great many, but several key studies that indicate in humans, by epidemiologic evidence, that one of the major causes of the increases in autism spectrum disorder and ADHD is likely to be certain pesticides, and they're very familiar pesticides. And sort of importantly, and this goes back, how we doing? Got plenty of time? 5 minutes, terrific. I think importantly, the best evidence we found is in the pesticides that have been around a long time. So our pyrethroids, some of the organochlorines, and organophosphates, pyrethroids, there was fairly strong evidence. For the newer agents, such as the neonicotinoids, there's less evidence. And what this said to me and to Dr. Roberts is that's an artifact of this pesticide progression. And I've been in pesticides since 1972. And back then, we basically only had to worry about the organochlorines such as DDT. And as we've watched every generation come into use, we start to see the next generation causing problems. So this idea of progression of pesticide agents being somehow a solution, it clearly is not. It's clearly a lack of time to actually observe the effects of the pesticides. And so there's no solution in going to the next round of pesticides. The solution is to not have the pesticides. So you can read the paper. You can see all the details. There's some nice tables and many references, but this is the framework for the paper. And it was a lot of fun to do. And we're really pleased that it was published in a really good journal. Thank you very much.
Parkinson's disease, ADHD, ASD, whew. But that's why we're here, right? So we can make a change in this. And so Dr. Chen is next, our home speaker from Mount Sinai.
Slides loading, so I just want to thank the organizers for having me to present. I'm not an advocate, I'm a researcher, and I'm a molecular epidemiologist. So my laboratory has been really using genetic tools. So as the previous speaker said, association is not causality, and so we fully know that as traditional epidemiology, that has been a real challenge because we are looking at all kinds of associations. So, and my laboratory has been using like molecular, especially genetic, epigenetic tools to actually find the causality. So, and not only that, sometimes we do use animals because the beauty of using an animal model is you can experimenting and then hopefully hone in on that causality issue. So today, I'm just going to present some of the results are preliminary and results on glyphosate. So I know it's a very hot topic these days, and so hopefully my results will shed some light on some other issues we should look at. So okay, first one. So what is glyphosate? I'm going to go through the background very quick for this group. I don't think we need a lot of introductions about what glyphosate is. It's basically, it's discovered in the 1950s by a Swiss scientist, Dr. Martin. And in the 1970s, it was discovered by the scientists in the Monsanto that this chemical can be used as a herbicide. And so it was subsequently patented as a herbicide at Roundup. That's probably the most well-known, and by Monsanto. And so in the 1996, there was the first introduction of the genetically modified glyphosate-resistant crops, and that was introduced to the market that has a profound change on the use of the glyphosate. So now, because there are crops that is, you will not be killed by the glyphosate. And so in 2000, so this, the Roundup, I think the glyphosate is out of patent. And so we all know in 2015, so the IARC, which is the cancer agency for the WHO, classified glyphosate as a probable carcinogen, so there's class 2A. And so there's a lot of controversies from, since then till now, up to now. And so in 2018, so the European Union actually reevaluated the use of glyphosate, and it was approved for limited use for 5 years, 5 additional years. I think it's going to be 2023. It's going to be reevaluated again. And so since the introduction of the GMO crops, so glyphosate is the most widely used herbicide in this country. It's probably in the world. It is, I think it's the most used herbicide that probably we've ever seen. And so how does glyphosate work? So it's actually a quite genius molecule. So it targeted this pathway, it's called the shikimate pathway. That is basically, you don't have to know all the details, but it's basically play a significant role in the synthesis of the 3 amino acids on the bottom, so thus to synthesize the proteins. So what is so genius about it is that this pathway exists in plants and not in mammals. So that's why, so the glyphosate actually blocks this pathway, so it stops the protein synthesis or amino acid synthesis. So that's why it kills the plants. So it has to kill the growing plants, and since it has active protein synthesis, but not only the plants has it, many of the microbes also has this pathway. So that's why I underline that, and then you're going to see why that is important. But we humans don't, and mammals don't. That's why it's considered safe for humans. So that's how it works. And so this, I think, is very similar to Virginia's slide. You see the next one. So before, this is in 2092. So before the GMOs, you see the use of glyphosate. It's, people use it, but not to a large extent. But this is what the usage now. And so these are the use of glyphosate throughout the years, and then you see there's a really sharp increase of usage, just because most of the crops, many of the crops in this country are glyphosate resistant. So, and then this talk is only focused about human health, so it has profound effects because of glyphosate. Also, not only kill plants, but also can modify the microbial community. So that has a profound effect on like the soil, which I'm not going to talk about. But the glyphosate these days has been present ubiquitously in the food chain. So in pretty much, starting with the crops, and then use it as not only the herbicide, they also use it as a desiccant. So actually it was sprayed before planting, during planting, and pre-harvesting. And because it seamless, it looks very harmless, so people use it a lot. And so, of course, it end up in the food chain. So is there a way to use the mouse? Yeah. Do they see the mouse there? No. There's no mouse here. Okay, but oh, here. How do I use this? Do you know how to use this one? Oh, never mind. But basically, you see on the right hand side, it's like it's in the farm animals because of the animal feed, and they end up almost like many of the food that not only in the like cereals. Now the latest is like wine, beer, and you name it. In the baby food. And also in tap water, like in rainwater, they tested it's basically ubiquitous. And then they have detected a trace amount of glyphosate. So, and then these are from a latest review article. I think Dr. Taioli is probably here. Yes, so I steal it from your paper. So she actually wrote a really nice review article about the exposures of glyphosate in the human population. So she has two nice graphs. One is an occupational population. So this is in the general public population. So they're from various of studies, I think across the globe, not just the US, from Europe, and then you see there's some of the one, if you can read the legend, so the glyphosate, this is in the urine. So they detected not only in adults, also in kids. So you have detectable trace of glyphosate. So one thing I want to point out is before, so like if you historic samples, so there are many samples that test negative. You cannot find trace of glyphosate. But now, like that percentage of urine samples that has a detectable trace of glyphosate as sharply increasing is almost like every samples that you test, you can find glyphosate in it. But again, being exposed doesn't mean it's harmful. So I have to say the level that you detect in urine is not very high. It is quite low, but it's there. So then the key question is glyphosate carcinogenic? So that's a huge, they generated huge debates, and we all heard about this lawsuit recently. Mr. Johnson was awarded $280 million, by the lawsuit against Monsanto, and then there's another one very recently, also was awarded close to $80 million. They're all from California. Both gentlemen had non-Hodgkin's lymphoma, and so it's like they generated like the public outcry, and of course, the stock market plummeted for the Bayer, who is the parent company of Monsanto. But in the scientific and regulatory community, whether glyphosate is carcinogenic, it's not a clear-cut issue. It's highly debated, depending on who you ask. And so, like IARC basically classified it as a probable carcinogen. So, but the EFSA, who is the European Food Safety Agency, conclude that it's unlikely to be carcinogenic. So I'm just highlighted here. They basically have the same data. They probably reviewed it differently by different community, and they reached drastically different conclusions. And then the US EPA also said it's unlikely to be carcinogenic. So, but what I'm arguing is we keep forgetting that cancer is only a small part of human health. There's many other health outcomes, so we should look at, and it has been ignored by the community. So, and here I found there's a knowledge gap, especially regarding to glyphosate, because it's such an emerging chemical that the use has been like sharply increasing and is still increasing. And so first of all, there's not many systematic studies on the non-cancerous outcome. And as if like everybody's afraid of dying of cancer, but nothing else. But there's many other health outcomes we should really look into. So I don't know whether they are harmful in the other outcomes. And then it's very few study looking at glyphosate and in the developing children. So most of the studies are on adult. And so there's even less study on the in utero or early exposure, like in the early childhood. And then we don't know about whether there's cross-generational effects of these chemicals. And we don't even know what is the dose response, because I think many of you know the endocrine endocrine disruptors, so like there's no good linear dose. So then that proposed a challenge of how you're going to study the toxic effect of glyphosate. So I'm going to present our study. So this study is in collaboration with Ramazzini Institute, who's in Italy. And so their expertise is the animal models. So we worked with them a lot previously. And so I started off working on breast cancer and endocrine disruptors and in the personal care products. And then we had a BSERC project. So we studied on the window susceptibility. And so this study was carried out in Ramazzini. And so what we decided to do is we're fully aware of the limitations of translating a rodent to a human. But this sprague-Dawley rat model is like the most well-known rodent models, and then it's really probably one of the best to recapitulate the human population. We actually use outbred rodents, not inbred. And we know, like, in terms of reproduction, they are very highly mimicking humans. And then we decided we're going to really go directly into the low dose. We're not going to do the high dose, extrapolating low dose. So we actually use the US ADI, so it's allowable daily intake, so which is 1.75 micrograms per kilogram per day, and it's considered a safe dose, so that's what we directly use. And we are fully aware at such low dose, we probably will not see much of a gross outcomes. So, and then here's basically the study design. So we actually expose the dams. So at the 6th day of gestation, we keep exposing them, and then the pups born, and then they were kind of breastfed by the dams. So during this period, we're not sure whether the pups are exposed, because we don't know whether glyphosate actually crossed placenta. And then even through the nursing period, we don't know whether they're exposed. So we don't know that. And then, after the weaning, so they just having drinking water. So I'm just going to go through quite quickly. So as I said, this is the dose. This is 1.7 milligrams. So it's the US ADI. And then we also use Roundup. So this is very important. The glyphosate and Roundup are two different beasts. And so, but in the Roundup, we calibrated, we use the same amount of glyphosate in the Roundup. So that's how we use, and through the drinking water. Oh gosh. Okay, so I'm going to go really quick. So then the first thing that we look at is the gut microbiome. So that's why in the beginning we actually highlighted the microbes. So we all know a gut microbiome has a profound impact on an array of health outcomes. So what we see is, just so you have to trust me, I have to go, I have 5 minutes this here. So, and we actually see significant changes in the microbiome in the pups, not actually in the dam. So this is very important. The dams, I think you have some changes, but not as profound as in the pup. And so that's one thing. So we basically look at an array of health outcomes. So this is one of the interesting findings. So we find the glyphosate and Roundup in the females has a significant delay of the first day of estrous cycle. So that's indicative there's something wrong with the hormones, with the estrogen. So there's a delay. So unlike most of the other environmental chemicals, the effect seems to be the opposite. So usually you find early estrous cycle. So it's like estrogenic, like BPA, But we found actually delay in both Roundup and glyphosate. And then when you look at the anogenital distance, so that is considered controlled by testosterone, you actually find increase age. It seems like this chemical is probably androgenic, which is probably, we haven't found any other chemicals that is, environmental chemical that is androgenic. So that goes to increased AGD. And then also when we look at the liver transcriptome, and then you see there's some changes in the transcription gene patterns in the liver. And then last is we look at the non-targeted metabolome. So this is taking advantage of our fantastic Exposome Institute. We did metabolome profiling. This is non-targeted. And then you see so the two panels, on the left side is female, on the right side is male. So you don't have to read the details, but basically what we see, so the red ones are the pathway that's dysregulated by glyphosate, I think. I can't even read. But the blue one, I think, is by Roundup. So the take-home message is there are certain pathways that are disrupted by the glyphosate and Roundup are the same, but there's distinct patterns. For example, the blue ones, that you only see it in the Roundup, and then the red ones, you only see it in the glyphosate. And then there are blue and red ones, they are common ones. So, so this slide to show that these two compounds, or the two two things are not the same. So that is impose a significant challenge of toxicological studies, because here is so that I said glyphosate and Roundup are not the same. So actually, if you have most of the people use Roundup, they don't use glyphosate. So you find all these chemicals. So has anybody can guess which one is glyphosate? Or which one you think is it's absolutely the worst one? Probably people will pick the one that is very complicated ones. But actually, you see this is a glyphosate. It's only 18% of the Roundup. So all the others are additive. And why that is important? Because there are many studies to show that it's actually glyphosate itself has the least biological effect. So you don't have to know what the test is. But basically, you see the Roundup actually consistently have worse effect than the glyphosate. So we don't know whether glyphosate is actually the culprit or is the additive. Okay, I have 1 minute to go. And, but the challenging thing is the formulation is different depending on which brand you buy. And the additive is different, and they're not always known. So they are not obliged to reveal what is their additive is in their products. So, okay, just to conclude, so I think my presentation hopefully to show that it's like a very urgent issue to study the effects of glyphosate in the other non-cancerous outcome, especially in children. And so now my colleagues at Ramazzini, actually they did a cloud sourcing to actually launch this called Global Glyphosate Studies. It's still a rodent study, but it's to study the life course of glyphosate, all the way till the end of life of the rodents. So hopefully, not only we can focus on the early developments, we can also see the life course risk. And then again, ultimately is how to translate the findings, how to do a systematic study in the human populations that actually capture all the potential health effects of glyphosate. So yeah, so this is the lab, and Brian who did the microbiome study, and Karina is the postdoc in the lab, and Lauren Patrick did the metabolome analysis, and of course Shanna, and who is an expert on the AGD, and then the people on the right are the team from Ramazzini. Thank you.